Good morning and welcome. With Research and Postgraduate Department of Chemistry, St. Thomas College, Pala, is conducting this webinar on viral diseases, challenges, and solutions. Every one of you know that this present scenario is an unprecedented one due to the over influence of COVID-19. In this context, this topic is very much relevant. Our topic for the seminar is viral diseases, challenges, and solutions. A resource person is Dr. Juby John, Senior Scientist and Assistant Professor of ACSIR Organic Chemistry. On behalf of Department of Chemistry, St. Thomas College, Pala, I welcome you all to this webinar. Thank you.
Through body fluids. There are two types of herpes viruses, herpes virus 1 and herpes virus 2. The symptoms of herpes virus infection include watery blisters in the skin or mucous membrane of mouth, lips, nose, or genitals. And the HSV1, the herpes simplex virus 1 infection, is mainly recognized by the appearance of painful ulcers around the mouth area. It can also cause infection in eye, brain, and also infection in babies. And herpes simplex virus 2 is characterized by the presence of painful ulcers on genitals. And here again, I want to emphasize that there are no drugs which can cure this virus infection. Again, coming to the next one, which is Ebola, and we all have heard about this disease due to the periodic outbreaks in West Africa. The most and the worst outbreak which was reported came in the period 2014 to 16 in West Africa. And during that time, 50% of the people who got infected lost their lives. It's quite an alarming mortality rate. So the Ebola virus is an RNA virus. It can cause a severe and fatal hemorrhagic fever. Hemorrhagic fever means you will have bleeding. The signs and symptoms of this viral infection starts between 2 days and 3 weeks after contracting the virus, and the symptoms start with the fever, sore throat, muscular pain. It progresses to vomiting, diarrhea, and at the same time the liver and kidney will show decreased functioning. And at this moment, some of the patients will start to bleed both externally and internally. And again, I have to say that there are no drugs which are there to cure this disease. But recently, a vaccine has been introduced for Ebola virus, but the efficiency of the vaccine is not yet published. The next one is MERS. MERS, most of you might have heard about it due to the COVID virus, because both MERS and the coronavirus, they both belong to the same family of Coronaviridae. This virus, the full form of MERS is Middle East Respiratory Syndrome. It was first reported from Saudi Arabia in the year 2002. And this is an example for a zoonosis. And most of you might know about zoonosis. A zoonosis is a disease which gets transmitted from an animal to a human being. We first came to know about a zoonosis after the Nipah virus infection, isn't it? So MERS is also a zoonosis. And once a person gets infected with MERS virus, it slowly develops into acute respiratory illness, same as in the case of COVID. And this can lead to a death of 40% of the people who are infected with this virus infection. And again, I am saying that there are no drugs or vaccines for this virus infection. And we all hear about the next one, Japanese encephalitis, almost every year due to the periodic outbreaks in the northern part of our country. This virus is transmitted by mosquitoes of the type Culex. Culex type mosquitoes are transmitting this virus. And normally, this virus infection comes and goes as a normal viral fever. But in some cases, symptoms like headache, vomiting, will come, which will slowly progress into confusion and seizures, and finally into inflammation in the brain. And the problem is that there is no treatment for this viral infection. Only the symptoms can be treated. And another problem is permanent problems, neurological problems are reported in the children who get cured from this viral infection. Now there is a vaccine for Japanese encephalitis, and I hope that our government will make it as a compulsory vaccine for at least children in the northern part of our country. And I will talk about dengue and Nipah in the coming slides. So let us talk about HIV in detail. So at the close of 2016, there were an estimated 37 million people living with HIV worldwide, with approximately 2 million people newly affected in 2016 alone. The introduction of HAART, highly active antiretroviral therapy, has transformed this viral infection from a lethal form to a manageable chronic condition. That is, if a person is getting HIV virus into his body, with the newer medications, that person can live a normal life for 20 years or more without much problems. It doesn't mean that you should get the virus, but I'm saying that there are very good treatment options presently. And most of you might have heard that early this year, 2 persons were cured from HIV with the new treatment options, which is a very remarkable news, but more trials has to be done on it. And unfortunately, our country is having the 3rd largest number of HIV patients in the world. And in this slide, I have shown some of the drug molecules which are currently in use. I don't want the students to get scared by looking at these structures. Just consider these molecules as small organic molecules that we take in when we have some infections. And all of these are available for us. These are the newer medications which are available. And now we have ritonavir and lopinavir also in India, but the rest are very good anti-HIV drugs. And the point that I want to stress here is that none of these are available in our country. Coming to the next one, which is hepatitis. Worldwide, viral hepatitis is the most common cause of liver inflammation. And in particular, types B and C lead to chronic disease in millions of people, and together can be considered as the most common cause for liver cirrhosis and liver cancer. There are 5 types of hepatitis, viral hepatitis: A, B, C, D, and E. Hepatitis types A and E are transmitted by the ingestion or intake of contaminated food or water. In the case of hepatitis B, C, and D, they are transmitted by the contact with infected body fluids, such as by receiving contaminated blood or blood products, by undergoing medical procedures with contaminated equipment. And it is also reported that hepatitis B can be transmitted from mother to baby during birth, from family member to child, and also by sexual contact. Nowadays, hepatitis B is also getting transmitted by the use of intake of drugs with contaminated syringes. And these, the above ones, the drugs which I have shown above are the ones which are currently in use for the treatment of hepatitis B. And tenofovir is a molecule which was developed by the professor which have worked in Belgium. And birinapant is the newest drug which is available for the treatment of hepatitis B. And I want to say that this is not available in our country. And when it goes to hepatitis C, these are the 2 molecules which are available now for the treatment in our country. And if you look at the molecule sofosbuvir, this is a very recent introduction as a drug. And the point here is that this molecule, this drug is very expensive. In the United States, one treatment regime of this molecule with this drug, it costs close to 1 crore rupees. In India, the company which developed this drug, Gilead Pharma, they have introduced a generic version of this drug which still costs 20,000 rupees for one treatment regime, which still is quite expensive for our Indian population. And these are the newest drugs which are available for the treatment of hepatitis C. And again, I want to say that none of these are available in our country. Coming to dengue, we are all familiar with this one, isn't it? So dengue fever, as you all know, is a mosquito-borne disease, and these are transmitted by mosquitoes of the type Aedes. The symptoms of dengue fever typically start between 3 to 14 days after the infection, and in a small proportion of cases, the disease develops into life-threatening dengue hemorrhagic fever or dengue shock syndrome. In the case of dengue hemorrhagic fever, what happens is bleeding, internal bleeding occurs, which results in low levels of blood platelets and blood plasma leakage. And in the case of dengue shock syndrome, what happens is dangerously low blood pressure is seen. It is estimated that around 50 to 500 million people are infected by dengue virus every year. And the problem is that there are 5 serotypes of dengue: dengue 1, 2, 3, 4, and 5. And developing a vaccine which is effective against all the 5 serotypes, it is the most major hurdle. And again, there are no antivirals that can cure this dengue virus infection. Whenever we have a dengue virus infection, what we do is we only treat the symptoms. That is, if you have a temperature or body pain, you take an antibiotic or an analgesic. Okay, coming to Nipah. I'm quite sure that we all came to know about this after the first outbreak of this viral infection in 2018 in our state. And I want to emphasize that it was only, we are all alive here and healthy because we live in Kerala. And we have to appreciate the health machinery in our state, because otherwise, why I'm saying this is like the transmission mode of this virus, it's very bad. It's like from 1 person it goes to 3, from 3 it goes to 15, from 15 it goes to 225, and from 225 it will go to thousands. And with our efforts from the government and health machinery, we could stop it at the 15 stage. The number 15. So in Kerala, a total of 19 people were infected and 17 lost their lives. The mortality rate was close to 90%. Yeah, it is such a deadly disease. The thing is that the Nipah virus infection in humans, it has a range of clinical presentations. Since some people, few people, there won't be any symptoms at all, but in some cases it can develop into an acute respiratory syndrome and fatal encephalitis. The problem is that slowly it will show, in some people it will show symptoms as a normal fever, and in the first case that was reported in Kozhikode, the doctor was very much well informed. He had read about Nipah virus infection 2 weeks ago before he treated that first patient. And as the patient was showing seizures and he had signs of encephalitis, the doctor thought that it might be a Nipah virus infection, and he sent the blood for testing. And somehow his timely intervention was the only reason that we could restrict the disease progression or disease transmission. And the main problem with this Nipah virus infection is that it will start with a fever, cough, headache, and it will worsen into a vital encephalitis or coma over a day or 2. That is, even if you feel that it's a Nipah virus infection and you take the blood for testing, as soon as you are getting the result, you will already be in a coma. That much fast this virus infection progresses. And again, I have to say that there are no vaccines or specific treatment for this viral infection. And you all know that a fruit bat is the natural host of Nipah virus, and it can go, the transmission can go through the fruits. When the bats eat the fruits, it can transmit to the fruits, and halfly eaten fruits, if human takes it, it can go to the humans and create a disease, or it can transmit viruses to pigs, and from pigs, from the meat, it can go to humans. The first reported Nipah virus infection was in 1996 in Malaysia, and it was transmitted from pigs to pig farmers. And during that time, they had to kill 1 million pigs to prevent the transmission of this virus infection. So we have heard that bats are good reservoirs of viruses. Why can it be like that? The recent research has shown that as soon as a virus enters the body of a bat, the immune system quickly acts and produces interferons. Interferons are signaling proteins which are produced by immune system. And these proteins, they inform nearby cells saying that there is a virus which is going to attack, and all the cells will be ready. The difference between we also have interferon alpha in our body. The difference is that the production of interferons in bats is lightning fast. That is what the researchers have said. It is lightning fast in comparison to that of humans. And another point is that even if the interferons are produced, bats won't have any symptoms, like they won't have an elevated temperature, they won't have any body pain or inflammation or anything, because as soon as they detect a virus, they will produce interferons, and at the same time they will also produce anti-inflammatory cytokines, so that even if they have the virus, they won't have any symptoms and they won't have any disease. The virus will just stay in their body. And the thing is, the bats won't kill the viruses. That is the second point. What the bats will do is that they will give the viruses all the necessary conditions so that the virus will stay in the body of the bats and mutate into the most lethal form. And then it will come out. Once the virus comes out, it will be in the most deadly condition. So we have to be aware of the bats. So coming to corona. I'm sure that everybody is an expert now in the COVID-19 disease. So the name corona, it came from a Latin word which means crown. Coronaviruses are RNA viruses, and these viruses belong to the family Coronaviridae. Normally, a coronavirus infection comes and goes as a normal viral fever by just having very mild symptoms. But the rarer forms such as SARS, MERS, or COVID-19, these forms can be very lethal, resulting in a respiratory infection or pneumonia and gastrointestinal infections leading to death. Coming to COVID-19. This is also known as SARS-CoV-2 or Severe Acute Respiratory Syndrome Coronavirus 2. This is transmitted through air via respiratory droplets from coughs and sneezes. That is why we have to maintain social distancing of at least 2 meters while talking. And also we have to use masks. And this is also an example for a zoonosis. That is a disease that is being transmitted from an animal to a human being. And it has been found that this disease was spread from an anteater of the type pangolin from the Wuhan market to a person who was selling it. And from there, it all started. And it is being said that now, as per today's stats, More than 12 million people are affected with this viral infection, causing the death of 5.5 lakhs. Now, let's have a small look at the mortality rate and age. I have taken this data from the New York City Health as of it was reported on May 13th. And as you can see here, you can see that for the age groups from 0 to 44, the people who get infected, 4% of the people who get infected lost their lives. It means that the younger age are quite safe, but not completely safe. And for the people who are above 75%, 50% of the people who got infected lost their lives. And what are the main reasons of these deaths due to COVID-19? The main reason is the pneumonia which occurs as a result of the COVID-19. The other recent reasons that were introduced are, the first one is cytokine storm. Cytokines are proteins which are produced by our immune system in response to an infection. So what cytokines do is, cytokines go to an infected cell and cause inflammation and make that cell inactive or lead it to death. And what does cytokine storm mean? It has been found that in some of the COVID-19 patients, the cytokines are produced uncontrollably. It is getting produced in mass numbers, and at this point, cytokines will not only infect the virus infected cells, but also the normal cells, and it will infect all the internal organs and slowly lead to death. The next reason is pulmonary thrombosis. It has been found that from around the world in many countries, during the autopsy of patients who lost their lives with COVID-19 infection, it was found that thrombosis or blood clots in small veins in the lungs was seen. The reason for this blood clot was not known until now, but it was seen that this blood clot resulted in a decrease in the oxygen level in the lungs, and they needed ventilators. Slowly, fluid accumulation or pus accumulation occurs, leading to pneumonia and death. And finally, the other reasons are the old people with high blood pressure, diabetes, heart problems, and cancer. These can also lead to death due to COVID-19. So what are the treatment options presently there and approved in our country? 2 antivirals are currently approved, which are remdesivir and favipiravir. Both of them are viral RNA polymerase inhibitors. But for our community, remdesivir is quite expensive. For one injection, it costs around 5,000 rupees, and a patient will need from 4 to 5 injections. In the case of favipiravir, one strip of the drug costs 3,000 rupees. It's very expensive. Favipiravir is given to patients who have mild symptoms, and remdesivir is given to patients who are having very worse conditions. And there are many claims saying that remdesivir is not that active, but still, as these are the only options, we are trying it. And the Indian DGCI, the Director General for the drug approvals, they have given approval for these 2 drugs. And the next thing that is being used currently is dexamethasone. Dexamethasone is known anti-inflammatory agent and also immunosuppressant. Whenever there is a cytokine storm, inflammation occurs throughout the body. So people who are needing ventilators are being administered dexamethasone, and dexamethasone, it helps to decrease the inflammation in the lungs and other parts of the body. And it is being reported that the intake of dexamethasone has decreased the mortality rate. Another treatment option that is being used is the use of plasma from recovered patients. The thing is that whenever we have a viral infection, just like if you consider chickenpox, what happens is that your body will detect chickenpox and your body will create antibodies, and these antibodies will be there for your life. So when a person gets recovered from COVID-19, the person will have antibodies against COVID-19. So what is being done is, from the recovered person, the blood plasma is taken up and injected to an infected person, so that the antibodies will trigger the immune system and produce larger amounts of antibodies, and thereby decrease the infection. But there are conflicting reports that the plasma therapy is not that efficient as it is being projected. And finally, supportive care, that is, care is being given to relieve the symptoms of the virus infection. So as soon as the COVID-19 was reported in India, my parent organization, Council for Scientific and Industrial Research, we started working on this to tackle this virus infection in different ways, such as digital and molecular surveillance, where we take virus strains from different parts of the country and identify the strain of the virus, rapid and economical diagnosis. So most of our labs are now involved in diagnosing, testing the virus infection, and development of repurposed drugs for COVID-19. I was leading this activity here at CSIR NIST. We made 3 molecules which are being tested against COVID-19, and we will be giving them to pharma companies, the processes. The next one is the development of hospital-assisted devices and personal protective equipments. And finally, we have introduced a portal, Arogya Path portal, which makes sure that everything that is required for the treatment and everything that's required for the health machinery, the police, and all due to address the COVID-19 pandemic is available throughout the country. Now, I have almost come to an end to the first part of today's presentation, that is talking about the viral diseases. Now we will have a look on the second part, which is a project on the targeted development of an antiviral drug candidate. So in the previous slides, I have told that there are no drugs available. In many cases, I told that there are no drugs available to treat many of the viral diseases. What is the reason? There are 2 main reasons. The first one is selective toxicity. Whenever we take a drug or a tablet, what happens is it goes through our mouth, through our esophagus, it reaches our stomach, from the stomach, through the stomach lining, it reaches the liver, and from the liver, through blood, it reaches the whole part of the body. And then this drug has to enter our cell and selectively attack only the virus. That is the main problem. Selective toxicity. There are thousands of drugs which can kill viruses in lab, but when it is given into a human being, it will not only kill the virus but also kill the human being also due to the side effects. So that is the first thing. As a virus can only replicate and live inside a host cell, the selective toxicity is the first problem. The second problem is the fast mutation rate of the viruses. If you look at HIV virus or influenza virus, they mutate very fast. So if you develop a drug now, today, it won't be good enough after 6 months because the virus will mutate quite fast. This is also the problem which arises during vaccine development also. There are no vaccines for HIV or common influenza viruses because of mutations. So as you all might know, a vaccine is an inactivated virus. That is a virus which cannot cause an infection. So whenever you are being injected with the vaccine, our body will detect a foreign particle, the antigen, and our immune system will create antibodies. So what will happen is that antibodies will remain in your body for life. So whenever you are having, whenever HIV virus or any virus gets into your body, the antibodies will suddenly act and inactivate the virus. That is how a vaccine works. So in this case, what happens is what happens is, as the virus mutates very rapidly, and if we are developing a vaccine, after 6 months, this vaccine won't be good enough, because whenever the mutated virus gets inside our body, the antibodies won't recognize it, and it will cause infection. So these are the two major reasons why it is difficult to develop antivirals. First one is selective toxicity. Second one is higher mutation rates of viruses. So now I am going to talk about a project for the development of anti-HIV drug candidates. So the HIV virus, human immunodeficiency virus, it is an RNA virus. What it does is, when it enters into a body of human beings, it targets a particular type of cells called as CD4 plus cells or T helper cells. These are specialized cells of our immune system, which helps in the immune signaling of our body. What the HIV virus does is that they selectively go and attack these cells and go and use these cells for replicating. As time goes on, the number of CD4 plus cells or T helper cells in our body depletes in such a way that our immune system will be the worst. And this condition is called as acquired immune deficiency syndrome. At the beginning of HIV virus infection, we can't say it has an AIDS. It's only at the later stage when the
Dr. Jubi, your audio is not clear. We can't hear.
Hello?
Hello, audio is not clear.
Oh, okay, sir. Check here. അത് ഒരു മണിക്കൂർ കഴിഞ്ഞപ്പോൾ സിസ്റ്റം ഓഫായി പോയേ.
Tojo, what happened?
സാറേ സിസ്റ്റത്തിന് ചെറിയൊരു ഇപ്പൊ ശരിയാവും, ഇപ്പൊ ശരിയാവും. ഒറ്റ മിനിറ്റ്.
ഓക്കെ ഓക്കെ.
ഹലോ സർ, അവിടെ പവർ പോയതായിരുന്നു. സർ അവിടെ പവർ പോയതായിരുന്നു. ഇപ്പോൾ ശരിയാവും, ഇപ്പൊ ശരിയാവും. ജോയിൻ ചെയ്തില്ലേ? ഓക്കെ, തിരിച്ചു വന്നു. മനസ്സിലായിട്ട് വന്നു.
Hello, am I audible now?
Yes, sir. It's audible. Okay.
Can you hear me?
Yes.
Okay. I will continue.
Okay.
I'm very sorry for the interruption because my power got lost. I'm very sorry. So I will continue without delay. So, what I was talking was how the HIV infection slowly progresses to AIDS, the situation of AIDS. And now, I'm going to talk about the initial stages of drug development. So what are the initial stages? So the first stage is like, you need to identify a drug target. You need to know a drug target, a stage of the virus replication where you want to block. So the drug targets are normally proteins, enzymes, or nucleic acids. So for this, what we did is, in order to find a drug target for HIV, what we did is, we looked at the replicative cycle of HIV virus. And it has 7 stages. In the first stage, the HIV virus, it attaches to the host cell, the cell membrane of host cell, and the genetic material is released into the host cell membrane, through the host cell membrane inside. The second stage is reverse transcription. During this stage, the HIV RNA virus is being converted into a new DNA. The RNA is being converted into a new DNA. And in the third stage, the new DNA is being incorporated to the host DNA, thereby forming a hybrid DNA. And in the fourth stage, this hybrid DNA, during transcription, it is being broken down into small mRNAs. And during the fifth stage, that is translation, the mRNAs are broken down again into polypeptide chains. And during proteolysis, that is the sixth stage, the polypeptide chains are broken down into smaller fragments or amino acids. And finally, new virus forms are thrown out of the host cell, that is budding out. New virus particles are being eliminated. So we decided that we would look at reverse transcription stage, where an enzyme called reverse transcriptase, it helps in the production of a DNA from the viral RNA. And hence what we wanted to develop was HIV reverse transcriptase inhibitor. There is one added advantage of targeting this enzyme, because both HIV and hepatitis B has reverse transcription in its life cycle. So any molecule which is a reverse transcriptase inhibitor can be used to treat both HIV and hepatitis B as the molecule tenofovir. It is a blockbuster drug which is being used both against HIV and hepatitis B. So the first thing was that we wanted to identify a target, and we have identified the target as HIV reverse transcriptase. So as you all know what an enzyme is. An enzyme is a substance which helps in increasing the rate of a chemical reaction inside a biological system. And if you look at this structure, you all know what an enzyme substrate complex is. Actually, this blue part is the enzyme, the white part is the active site, and this pink part is the enzyme. And you can see the grooves on these, the enzyme and the substrate. And you might have heard, you have learned about the lock and key principle. Actually, what these grooves means is that they are functional groups. So on the active site of the enzyme, there will be functional groups. And on the substrate, the functional groups will be complementary to that of the functional groups which are present on the active site, so that these functional groups can be hydroxyl group, amine group, acid, halides, it can be any functional groups. And whenever these come together, these functional groups interact and form an enzyme substrate complex. So what we need to know is that to design an enzyme inhibitor, we need to know the functionalities present on the enzyme. And what is an enzyme inhibitor? An enzyme inhibitor is a molecule which can bind to an enzyme and decrease its efficiency or activity. Two types of inhibition are reported. One is reversible inhibition and irreversible inhibition. Now, let us look at the basic units of DNA and RNA, because we are going to talk about reverse transcriptase, which is an enzyme which helps to make a DNA from an RNA. So we need to know how a DNA and DNA is being formed. So the basic units are nucleobases. There are two types of nucleobases, which are purine bases and pyrimidine bases. Purine bases are adenine and guanine. Pyrimidine bases are thymine and cytosine. And in DNA, we have four nucleobases, which are adenine, guanine, thymine, and cytosine. And whenever a sugar is attached to this nucleobase, it becomes a nucleoside. And when one hydroxyl group is missing from the pentose sugar from the 2' position, it will be called as a deoxynucleoside, deoxy sugar. Yeah. And whenever a phosphate group is attached to the 5' position of the nucleoside, then it will be called as a nucleotide. So the basic unit is a nucleobase. When a sugar is attached, it is called as a nucleoside. When a phosphate group is attached, it is called as a nucleotide. So what will happen? How is the DNA strand being formed? So what happens is, to the nucleoside, to the 5' position, 3 phosphate groups are being attached. It is being done by enzymes called as phosphoryl kinases. Then what happens is that of a strand, a hydroxyl group of a sugar moiety, it attacks the phosphorus atom and then kicks out the diphosphate group and becomes a part of this strand. And 2 strands with complementary bases, they come together and form the DNA. And in this, complementary strands will come together in such a way that adenine forms 2 hydrogen bonds with thymine, and cytosine will form 3 hydrogen bonds with guanine. Okay, and this is how a DNA strand is being formed. So the first step of the drug development is identification of the target. Our target is reverse transcriptase enzyme. Now what we want to know is, we have to know the functional groups which are present in the active site of reverse transcriptase enzyme. For that, only then we can design a molecule which can selectively bind to the active site of the enzyme and inhibit its activity. So for that, what we looked is, we looked at the complex of this deoxythymidine. This is a building block of DNA. We looked at its binding with reverse transcriptase enzyme and found that 3 major parts are required for an effective inhibitor. The first part is a triphosphate moiety or a mimic, and this triphosphate moiety, it chelates with 2 metal centers of the reverse transcriptase enzyme. There are 2 magnesium cationic centers present in the enzyme. It is a metalloenzyme. Reverse transcriptase is an example for a metalloenzyme. Then it needs a nucleobase. It needs a nucleobase, and this nucleobase, it engages in Watson-Crick base pairing with the template primer of the enzyme. And finally, it needs a linker which connects both the nucleobase and the triphosphate moiety. So 3 portions, 3 parts should be there in the inhibitor, which is a triphosphate moiety or a mimic, then a nucleobase which can engage in Watson-Crick base pairing, and a linker which attaches both this nucleobase and the triphosphate moiety. So what happens is, for any moiety to interact with the reverse transcriptase enzyme, it has to go a triphosphorylation, which is done by phosphoryl kinases. Sidowoodin is the first molecule which was approved for treatment against HIV. It undergoes a triphosphorylation before it can interact with the reverse transcriptase enzyme, as shown here. The next is tenofovir. Tenofovir has already one phosphonate group, so it only has to undergo phosphorylation twice. So what we thought that, would it be advantageous to avoid these phosphorylation steps so that the molecule that we designed, when it reaches inside the host cell, it can directly attach to the reverse transcriptase enzyme and inhibit the activity, thereby avoiding this triphosphorylation. What we did is we looked at the structure of these two active molecules and came up with a structure where a carboxylic acid group was attached alpha to the phosphonate group, and we thought that this carboxyphosphate group will be an effective mimic for a triphosphate moiety. And this is what we did. So first stage was we identified a target. Second stage is we designed a molecule, designed an inhibitor. The third stage is you have to synthesize the inhibitor. For that, what we did is we did several organic reactions and we made molecules with the nucleobase, carboxyphosphate group, and we used cyclopentane and cyclopentene as the linkers. And what we next did was that we also made analogs with different nucleobases. In first, we used thymine, then we also used uracil, cytosine, fluorouracil, and adenine. So the next stage is we have to check the biological activity, the antiviral properties of these molecules. So we will come through the stages. The first stage was identification of the target that we did. It is reverse transcriptase enzyme. Second one is we have to design an inhibitor for this enzyme. We designed it. Third stage is we synthesized the inhibitor. And fourth stage is biological testing of this inhibitor. And during this, we found that this molecule, the molecule in red, that is the molecule with thymine, carboxyphosphate group, and the cyclopentyl moiety, it showed the best activity. That is in nanomolar level. It inhibited the HIV reverse transcriptase enzyme, and we were very happy. And the next stage is whenever you find that a molecule is active against any microorganism, you need to know the mechanism of action. So what we did is that we made a complex of our molecule with the reverse transcriptase enzyme, and we checked the X-ray crystal of this molecule. So how this is being done is like, so whenever you have a broken bone, whenever you have a fracture, what you do is you go to a hospital and get an X-ray of the portion. And there you can see that if the bones are intact, if everything is all right, or if there is a fracture, you can see through an X-ray. In such a way, if you get a crystal of a molecule, organic molecule, you can put it under X-ray. And from X-ray, you can see how atoms are bonded together. You can see how carbon-carbon is being bonded, how carbon-oxygen is bonded. In such a way, what we did is we made a complex of our molecule with the reverse transcriptase enzyme and checked the X-ray, and we found that the carboxyphosphate group was chelating with both the magnesium metal centers, just as in the binding of natural deoxythymidine triphosphate, which we have seen earlier. It was effectively mimicking the triphosphate moiety, and we found that our hypothesis was correct, and we developed a novel antiviral with therapeutic potential. So we will come through the stages once again. We had to identify a target, which was reverse transcriptase. We had to design an inhibitor, which we have done. Then we synthesized the inhibitor. And then in the fourth stage, we checked the biological activity of the inhibitor. In the fifth stage, we checked the mechanism of action. The next stage is structure activity relationship studies. In this, what we do is whenever we have an active molecule, we make small changes in the structure of the active molecule, and we look at the biological activity. So what we did is, this is our first molecule that we have a nucleobase, carboxyphosphate group, and cyclopentyl linker. What we did is, during structure activity relationship, we removed this methylene bridge of cyclopentane ring and made the linker as an acyclic one. It's an open chain structure. So we designed molecules and we synthesized it with by following different organic reactions, And we made molecules with different linkers such as butyl chain, butynyl chain, butynyl chain, cyclopropyl, cyclopentyl, cyclohexyl, norbornyl, and benzene ring also. In all these cases, it is an open chain. You can see that it's open chain. And we have synthesized it. And next what we did is we checked the biological activity. And at the first stage, we were very disappointed that all these molecules lost their activity against HIV viruses. But then we checked its activity against different DNA viruses such as cytomegalovirus that I have mentioned before, herpes simplex virus, and also varicella zoster virus. Varicella virus is the one which causes chickenpox. And we found that these molecules were showing excellent activity against these DNA viruses. That is, the activity has shifted from HIV, which is an RNA virus, to DNA virus during our structure activity relationship studies. And we found that this molecule with a butynyl linker was showing the best activity, and the nucleobase that we used was a thymine. And next what we did is that we changed thymine and made all other nucleobase connected molecules such as fluorouracil, guanine, adenine, and cytosine, and found that the molecule with guanine was most effective and most selective towards different viral DNA polymerases, and these molecules were inhibiting the activity of these DNA viral polymerases. And finally, from one work, we found that by a simple change in the structure, leads could be generated for RNA viruses and DNA viruses. And after coming back to India, what I have done is I have changed the nucleobase part for several heterocycles, and we have seen that hydrogen bonding is not the only thing that we need. We also can design molecules where heterocycles can act in a pi-pi interaction. Through pi-pi interaction, they can interact with the nucleobases with the pi-pi interaction, and it can show activity. And there is a lot of noise coming. Hello?
Hello, are mic on cheyidana. Antares, please switch off your mic. Antares, please switch off your mic. Sir, ignore it.
Antares, please. Mic.
Okay, it's okay.
Okay, so what we have found is we have found a molecule with the activity which has progressed to nanomolar levels. We are very excited in this result. And now what we are doing is we are again doing structure activity relationship studies, and we have reached a stage that we can go for prodrug synthesis. Prodrug synthesis, and it can start in animal trials. This is being done here in India now, and these are unpublished results. So what are the challenges that we are facing now? In our country, a major population is fighting against different viral diseases. And now we can see that India is the country which is having the third largest number of COVID patients, and it is going to go worse for sure. The main problem is that in many of the diseases that I have mentioned before, the most modern drugs which are very efficient are not available for our population because the multinational companies are not ready to sell these drugs in a cheaper price. Everything is very expensive, and they only are selling these in the Western countries. What I want the younger generation who are listening to me is that some of you, at least one or two of you, should take up science as their career and should work in such a way that we have many problems and we should tackle some of the problems. And what I want to say is that India is a country where we launched 104 satellites in one go. Then I'm quite sure that we can make affordable drugs for our people. And I'm sure that some of you will take up my advice and take up research. And finally, coming to acknowledgements, I have to acknowledge my director, Dr. Ajay Ghosh, Dr. K.V. Radhakrishnan, my HOS, and other scientists of organic chemistry section, students of organic chemistry, assistants with different characterization techniques, my collaborators from KLU in Belgium, and finally different funding agencies. And this is my research group. I cannot do anything without their hard work and enthusiasm. And before I stop, I want to leave this phrase for your thought. I want all of you to be a virus and to see the world. Why I'm saying this is that whenever we have a viral infection, what we do is we take up a drug so that we will inhibit the viral infection. But what will the virus do? The virus will mutate in such a way that it will overcome the effect of the drug. So this is how life is also. You will have a lot of problems, you will have a lot of hardships. You all should mutate in such a way that you should overcome the problem. And if you are mutating in such a way, you can overcome all the problems, all the hurdles, all the challenges, and then you can lead a very good life, and you can reach any goals and also reach any dreams that you have. Thank you very much for your patient listening, and I'm ready to take up all the questions and queries that you have, and I'm very sorry for the interruption that happened in between. Thank you very much. Thank you.
Thank you, Dr. Juby.
Yeah.
If you need any clarification, please ask. This is the time for clarification. You will get the time. If you need any clarification, please ask.
And kindly fill the feedback form. It's on the chat link.
Juby, I am Ranjit Thomas from SB College. Actually, I have a question for you. Shall I ask some questions?
Yeah, sure. Please go on.
Okay, so one of the first question is that, and how do you found that there is a pi stacking interaction between the drug and the enzyme, virus enzyme, and is there any possibility to tune the or to increase the interaction between the nucleobase and the drug? Okay, my first question.
Yeah.
Second question is that, in the first section you have told that the bats are having a special feature such that the viruses are masked in their body, and after some time, after mutation, they will come to the virulent form. At that time, will that virus kill the bat? Or it will infect the bat?
Okay. So the first I will answer the first question. So how we came, the pi-pi interaction is a hypothesis now. We are hypothesizing because in the heterocycles that we used, there were no hydrogen bond donors. So there was no possibility for hydrogen bonding with the nucleobases present in the enzyme. And we found that once the pi surface was increasing, the activity was increasing. And from these results, it is the only possibility, it's the only way that pi-pi interaction should be there. So we are hypothesizing that it is pi-pi interaction. And now what we have to prove it. So what we are doing is, we are trying to crystallize the molecule with the viral enzyme, and we will take an X-ray. And from that, we will be quite sure that it is a pi-pi interaction. I hope that I have answered your first question. Now coming to the second question, the viruses in bats, it gets mutated in the bats itself. So the immune system of bats are knowing that the viruses are mutating in its body, and it is always ready to prevent the infection from these viruses. Bats will never get infected from these viruses, even if it mutates to its most lethal form. It will only be a reservoir or a carrier of these viruses. That is that is an ideal condition. If humans have the same, if humans can adapt just like a bat, it will be the ideal condition, isn't it? So researchers are now actively working on it to find a way how these bats are overcoming these viruses without getting any infection. I hope
Thank
that
you.
I have.
Thank
Yeah.
you. Sure. Thank you very much.
If there is no any interaction, you can wind up.
So I have just a minute, Martin. I saw that
Okay.
there is one question here from Rajakumari Rajendran to comment on the bioactive components of plant-based drugs for COVID-19. So what I can say is that many people claim that they have found plant-based formulations or drugs against COVID-19. But I am quite sure that none of these have scientific proofs for its activity. But what I can understand is that in the case of COVID-19, what these plant-based drugs can do is that they can relieve the symptoms. They can help the symptoms. For example, there are many plant-based formulations which are anti-inflammatory agents. They can help to decrease the inflammation in the body. As I have mentioned before, COVID-19 can cause inflammation in the lung, thrombosis, and lead to pneumonia. These plant-based drugs can help to decrease the inflammation, and also there are many formulations which can boost the immunity, which will help to at least if we take up now, it will help to prevent the infection at least, or it will result in such a way that it will come and go as a normal infection. Otherwise, there are no plant-based drugs which can act as antivirals. That's what I'm stressing. No antiviral plant-based drugs are there. There are only drugs which can help or alleviate the symptoms. I hope that I have answered the question. And there is one question from Praveen Prakash saying can you please briefly mention the action of hydroxychloroquine? So the thing is that, as you know now, that the hydroxychloroquine is no more used as a drug for the treatment of COVID-19. What was proposed is that in the life cycle, if you have seen, in the case of HIV, you've seen that the first stage is attachment of the virus to the cell wall. So what chloroquine does is that it prevents the attachment of the virus to the cell wall of host cell. This is being done by chloroquine is a basic molecule. For a virus to attach to the surface of the cell wall, the condition should be acidic, and by changing this condition, it is hypothesized that chloroquine prevents the entry of the virus to the host cell. I hope that I have answered the question of Praveen. So there is one more question. Can I answer, Martin? Is it okay?
Yes, okay.
Okay.
You can continue.
So is the lifetime of these drugs related to its efficiency? How we can study? Yes, of course. So if we have a drug, if we have an efficient drug, it should stay as much as possible in our body. Yeah. Only then we can decrease the dose. I can switch to Malayalam also. So in the case of antibiotics or any other drug, or in case of paracetamol, we take it thrice or 4 times a day. It is because after some point, the whole external or the foreign bodies or chemicals, whatever it's there, it is being expelled by our kidney or our liver. So the thing is that for the effect of the drug to be there, we have to take up these drugs again and again. അതിനാണ് നമുക്ക് വീണ്ടും വീണ്ടും ഈ ടാബ്ലറ്റ്സ് അവർ കറക്റ്റ് ടൈം പറയുന്നത്. അത്രയും സമയമേ ആ ടാബ്ലറ്റിന്റെ നമ്മുടെ ശരീരത്തിൽ കാണത്തുള്ളൂ. സോ ഹൗ വി ക്യാൻ സ്റ്റഡി എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ നമ്മുടെ എക്സ്ക്രീഷൻ ഫ്രം അവർ യൂറിൻ ആൻഡ് അവർ ഫീക്കസ് വി ക്യാൻ നോ ഹൗ മച്ച് ഈസ് ബീങ് എക്സ്പെൽഡ്, എന്തുമാത്രം ഉണ്ടെന്ന് നമ്മുടെ എക്സ്ക്രീഷനിൽ നിന്ന് മനസ്സിലാവും, നമ്മുടെ ബ്ലഡ് ടെസ്റ്റ് ചെയ്യാമെങ്കിലും മനസ്സിലാവും. So I think that Sandhya, I have answered Sandhya's question. And then one question from Sudish: how we assume an initial structure of a drug for a new virus? Do we use computational studies? So the thing is that whenever what I am following is I look at the structures of already existing drugs, and what I do is I can do is I can play on the structures. As an organic chemist, I can design structures so that it will have a similar geometry as that of an existing drug, but the components will be different. Yes, we can use computational studies. Now, with simple docking studies, I'm quite sure that nothing will happen. There is a lot of docking studies going around in the world, and a lot of papers are coming saying that this drug will be good, that drug will be good. But what I have to emphasize is that not a single drug has come out from these docking studies until now. But there are very new tools for docking, which is very expensive. I know about one which costs around 10 lakh rupees per month for using this tool, but it is quite efficient, and a lot of drugs are being tested in the United States. Most of the cancers that we see now among us, it can be treated with the drugs which were formulated by using the most modern computational studies. In the case of designing an antiviral drug, computational studies are not good enough, because we have to take into consideration all the factors that it has to be selectively toxic. When we do a computational study, we will take a drug molecule and we will take an enzyme. If we see that it is docking quite good, we will say that it is good. But the thing is that it has to go through all the parts without causing any side effects. It has to enter the host cell and then it has to attack the virus. So in a part, computational studies can be used when we have an active molecule. When we have an active molecule, computational studies can be effectively used to alter the structure during structure activity relationship, to alter the structure in such a way that the activity can be increased. Otherwise, from scratch, I don't think that computational studies can be used, at least now. With the present tools that are being used in India, it's not possible. There are very expensive tools which we have to buy and which we have to use. I hope that I have answered Sudish's question, and there are no more further questions in the chat box.